Hello friends, today we are going to discuss one of the important theme in carbohydrate metabolism, that means mechanism of glucose oxidation energetics. Mechanism of glucose oxidation energetics. Carbohydrates are the major dietary source of energy, with glucose as the central molecule or the crux point for ATP production. All this can be studied under the one platform called carbohydrate metabolism. Carbohydrate metabolism. There are different pathways under carbohydrate metabolism, like glycolysis, that means EMP pathway, Krebs cycle, glycogenolysis, glycogenesis, gluconeogenesis, hexose monophosphate shunt or pentose phosphate pathway, uronic acid pathway, fructose metabolism, galactose metabolism. All these are different heads under the major head of carbohydrate metabolism. For instance, glycolysis, Embden-Meyerhof pathway, Krebs cycle, gluconeogenesis, glycogenolysis, uronic acid pathway, hexose monophosphate shunt, fructose metabolism, galactose metabolism. There are different pathways under carbohydrate metabolism, wherein the first two are the major energy producing pathways. They are the major sources of energy producing pathways: glycolysis and Krebs cycle through electron transport system. How? We'll discuss about this. We'll majorly focus on this topic, okay? Rest of topics like gluconeogenesis is nothing but the synthesis of glucose from non-carbohydrate precursors like amino acids, glycerol. Glycogenolysis, the name it implied, breakdown of glycogen to form glucose. Glycogen is synthesis of glycogen from glucose. Then uronic acid pathway is a conversion of glucose to glucuronic acid. Then it's a kind of direct oxidative metabolic pathway, just like hexose monophosphate shunt, otherwise called pentose phosphate pathway, TPP simply called. Here there is a bypassing of glycolysis and tricarboxylic acid cycle. That means directly the glucose is oxidized into carbon dioxide and water. What we call direct oxidative metabolic pathway is nothing but the hexose monophosphate shunt or pentose phosphate pathway. This under the same line, uronic acid pathway comes. Then fructose metabolism and galactose metabolism. And these are the major issues under carbohydrate metabolism, what we call simply the glucose oxidative energetics. It deals with these two pathways, okay? We'll discuss point by point how it works. Glycolysis regarding the basics of glycolysis. So it's called Embden-Meyerhof pathway. Meyerhof pathway. It is a universal pathway in all the living cells. All the living cells will undergo glycolysis. Even there are certain cells in our system, for example, retinal cells, erythrocytes, where there is no mitochondria. That means if there is no mitochondria, there is no possibility of Krebs cycle. If there is no possibility of Krebs cycle, there is no higher production of ATP. But in these kind of cells, glycolysis is a major source for ATP production. That means it is called universal pathway. And glycolysis is the main feeding source for brain as well. And how it works? For example, how it starts. There are three phases. For example, first the glucose becomes glucose 6-phosphate. This glucose 6-phosphate is impermeable to cell membrane. That means it's a what we call activation by hexokinase enzyme. That means ATP has been utilizing here. And this glucose 6-phosphate, what we call the first step or activation step, is an irreversible step. It's an irreversible step. Then glucose 6-phosphate become fructose 6-phosphate. Then this fructose 6-phosphate become fructose 1,6-bisphosphate. Here exam is phosphohexokinase. Here phosphofructokinase. And this fructose 1,6-bisphosphate, it breaks down into two. One DHAP, dihydroxy acetone phosphate. Its acting enzyme is aldolase. And glyceraldehyde 3-phosphate. This glyceraldehyde is simply called GAP. Okay. Then these are 3 carbons each. So far it is 6 carbon. Then it converts into, by means of an glyceraldehyde 3-phosphate dehydrogenase. Here it becomes 1,3-bisphosphoglycerate. Then again it becomes 3-phosphoglycerate. Phosphoglycerate kinase. That means here one ATP is produced, and the 3-phosphoglycerate become 2-phosphoglycerate, and this 2-phosphoglycerate becomes phosphoenol pyruvate, and acting enzyme is enolase, simply by removal of H2O, simply called PEP, and this phosphoenol pyruvate meant to become pyruvate, acting as an enzyme pyruvate kinase. Here another ATP will be produced. Then, okay, here also one more glucose molecule will be utilized. ATP will be utilized. ADP. Then here the most important component, NADH. NADH plus H plus will be formed from NAD plus. This is very important. Okay. That means is sometimes not only the glucose, the glycogen through glucose 1-phosphate, it becomes glucose 6-phosphate. So it's about the glucose metabolic pathway. That means we can say it's a sequential representation or sequential flow of step that converts glucose or glycogen to pyruvate or lactose with ATP molecule. Sometimes it's called lactose also. That means lactate under anaerobic conditions. It is under aerobic conditions. So the point is clear based on this. Glycolysis can take place both under aerobic and anaerobic conditions. If it is under aerobic conditions, if the starting molecule is glucose, the glucose simply becomes pyruvate. If it is under anaerobic conditions, the glucose becomes lactate. This is very important. Sometimes instead of glucose, the glycogen will be the starting molecule, and it converts to glucose 6-1-phosphate, then it becomes glucose 6-phosphate. Here 1 ATP will be saved. So we'll discuss about the end regarding its energetics. So that means the sequential flow, simply the glucose, the help of hexokinase. Here 1 ATP has been utilized to become glucose 6-phosphate. Then phosphohexoisomerase, which converts glucose 6-phosphate to fructose 6-phosphate. Then fructose 6-phosphate, again another ATP. Here 1 ATP is consumed. Here 1 ATP is consumed. Here second ATP has been consumed. Then it becomes fructose 1,6-bisphosphate. This through aldolase splits apart. Splits. Glycolysis. Lysis, breakdown. Here breakdown is with reference to fructose 1,6-bisphosphate breaks down to form dihydroxyestrogen phosphate, DHAP, and glyceraldehyde 3-phosphate. There are 3 carbon compounds. This is with reference to glycolysis. This is a very important point. So up to this phase we can say as energy investment phase. Energy investment. Because ATPs has been consumed here, and this is called split phase. Split phase. Then here ATPs has been produced. For example, 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. Here 1 ATP has been produced. And another thing, phosphoenol pyruvate, when become pyruvate, another ATP is produced. Second ATP. That means it is called energy generation. Energy generation phase. So broadly, it has 3 phases. 1, the energy investment phase. 2, the split phase. 3, the energy generation phase. It's about different compounds and how they form. That means ultimately leads to pyruvate. Okay, and most important point, as we have discussed, energetics. This is very important. These are all basics. We already dealt in the lower classes, but in advanced issues we have to learn regarding its energetics. How many ATPs are produced? In what way? This is very important. Say there is the enzyme glyceraldehyde 3-phosphate. 3-phosphate dehydrogenase. Here NADH plus H plus has been produced. That means 2 molecules, because here when fructose 1,6-bisphosphate splits into 2, that means dihydroxyestrogen phosphate, at the same time glyceraldehyde 3-phosphate, and they can be converted because there is isomerase enzyme. This isomerase converts, that means totally 2 more compounds and molecules will come from breaking of fructose 1,6-bisphosphate in the way of 3 carbon compounds. Therefore, the 2 NADH molecule, when this glyceraldehyde 3-phosphate, then become 1,3-bisphosphoglycerate, then 3-phosphoglycerate like that. Here the operating enzyme is glyceraldehyde 3-phosphate dehydrogenase. Here the first coenzyme, this is important, coenzyme, and this NADH will be, that means nicotinamide adenine dinucleotide will be utilized. It has 2 forms, oxidative form NAD plus and reducing form NADH. and this reducing form, what we call reducing element of nicotinamide and dinucleotide, what we call NADH, will be utilized in electron transport for the generation of ATP at the rate of 1 NADH equal to 3 ATPs. This is very important. What we call energetics, simply energetics of glycolysis. 1 NADH will generate 3 ATPs. Okay, as it was glycerol 3-phosphorylated, at this step 1 NADH has been produced. That means total 2, since there is 2 compounds involved. That means 6 ATPs. We'll discuss in when we discuss in electron system how the NADH, the reducing elements, and FADH, the flavin adenine dinucleotide involves. That means in electron transport system, and how they generate ATPs by what we call oxidative phosphorylation. Electron transport coupling with oxidative phosphorylation by a leaving motor system. Okay, we'll discuss later in the next class about the same. But here the thing is simply the regarding energetics. 1 NADH 3 ATPs, so total 2 NADH, the 6 ATPs. Then what we call substrate level phosphorylation. Substrate level phosphorylation. During the sequential flow of glycolysis, there are 2 ATPs have been produced. That means when 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. 1,3-bisphosphoglycerate become 3-phosphoglycerate. 1 ATP has been produced. Another step. When the phosphoenol pyruvate, what we call PEP, become pyruvate by means of pyruvate kinase, another ATP is produced. That means pyruvate kinase. One more ATP is produced. So total is 8 ATP. But this is not the case all the time. Here 6 ATPs. If NADH uses malate aspartate shunt, it produces 6 ATPs. If it uses glycerol shunt, it produces only 4. That means either 6 if it is 6 ATPs, if it uses malate shunt, or glycerol shunt, only 4 ATPs. So it depends. Then we have one more thing here, utilization. ATP utilized. That means here one actually 2, because 2 compounds will come out now. That means 10, but 8. Why I wrote 10? Because minus 2. In the way of ATP utilization at 2 steps. Say glucose become glucose 6-phosphate. Then one more time, fructose 6-phosphate become fructose 1,6-bisphosphate. Here 1 ATP is utilized and another ATP. That means actually out of 10, 2 ATPs has been utilized. When glucose become glucose 6-phosphate, 1 ATP has been utilized and through hexokinase, and when fructose 6-phosphate become fructose 1,6-bisphosphate through the enzyme phosphofructokinase, another ATP has been utilized. That means 2 divided 8. Actually since there are 2 rounds, so we have presented as 2. That means 2 rounds means 3 carbons in 2 times. So 2 ATPs to so 6 plus 2 8, 8 plus 2 10, and out of 10 2 has been utilized, so 8. Regard it is, but 8 is not constant. I told you already, it depends on the kind of shunt. If it is malate shunt, 6. If it is glycerol shunt, 4. So 8 or 6. It is simply energetics is 8 or 6 ATPs. It is the end point. That means If somebody asks you how many ATPs may be generated from one glycolysis, from one molecule of glucose, you have to say 8 ATP molecules if it utilizes malate shunt. These 6 ATP molecules if it utilizes glycerol shunt. This is very important. At the end we get 38 ATP molecules. Say malate shunt. 36 to reduce glycerol shunt. That means through the combination of Krebs cycle ATPs and the NADH, that means the reducing elements which are same in the electron transport system, will generate this. Here, except the substrate level phosphorylation where the ATP has been produced at the substrate level, the NADH, which are the important elements in ATP generation, how? They denote electrons. They denote electrons, and this electron transfer coupled with oxidative phosphorylation through the mediation of protons, we'll discuss later. Okay, this is how, for a basic idea I'm saying, okay, how they produce ATP. That means because of reducing elements like NADH and FADH2, they as electron donor through electron transport chain, they will produce ATP, because the ultimate here oxygen is an electron acceptor. Okay, then it's about energetics of glycolysis. Then the Krebs cycle, this is the most important point. Krebs cycle, that is named after Alfred Hans Krebs. Otherwise called citric acid cycle, otherwise called tricarboxylic acid cycle. Krebs. Hans Krebs is the first one who has propounded this mechanism. And citric acid, why? Because the first formed compound by the combination of acetyl coenzyme A plus the oxaloacetate is citric acid. Tricarboxyl, why it is called tricarboxyl? Because the first formed compound, that means say the citric acid, that means citrate simply, then cis aconitate, then the isocitrate, all are tricarboxylic acid. Since they form tricarboxylic acid, which are 6 carbon compounds, it is named after tricarboxylic acid. Since it is named after the Hans Krebs, because he is the one involved in that. Then the citric acid is the first compound. And one more important point regarding citric acid is a major source of ATP production in our cell. Around 65 to 70% of ATPs will be produced here. At the same time, 2/3 of oxygen is consumed here only. That means majority of the oxygen consumed, what we consume by our, that means what the cells consume will be utilized by the Krebs cycle, and the majority, that means 2/3 of ATPs, around 70% of the ATPs has been produced. That means there is a production point of ATP, the Krebs cycle. And the Krebs cycle will take place, unlike the glycolysis which takes place in cytosol, and glycolysis means both anaerobic and aerobic. If it is anaerobic, 2 ATP molecules, just 2 ATP molecules. If it is aerobic, 8 or 6, depending on the malate or glycerol shunt. If it is malate, 8, 6 glycerol shunt. But here the Krebs cycle it will take place in mitochondrial matrix. Will take place in mitochondrial matrix. Why? There is an important component. Because the electron transport system, I told you, which is a major source for ATP generation. The electron transport system the enzyme the same will be located at the inner mitochondrial membrane. Inner mitochondrial membrane. In the proximity of the same, the Krebs cycle enzyme should be present, so as to facilitate ATP through a process called oxidative phosphorylation. The generation of ATP is through oxidative phosphorylation. This is very important. And how the Krebs cycle will come? And the Krebs cycle is considered as, it's like high trophic point where different roads connect. That means the intermediate compounds of Krebs cycle is involved in one way or the other way with individual metabolic pathways. And this is both anabolic and catabolic pathways. Okay, I will tell you how. Regarding the first compound, acetyl coenzyme A. That means how first pyruvate, which we get in glycolysis, will become acetyl coenzyme A. Binding here the enzyme is pyruvate dehydrogenase. Here NAD plus becomes NADH plus H plus, same time CO2. addition of COA SH. That means called the sulfur bond. This is called oxidative decarboxylation. Oxidative decarboxylation. In Krebs cycle, simply what we call Krebs in the definition, it is nothing but the essential involvement, that means the conversion of acetyl coenzyme to carbon dioxide and water. Simply the acetyl coenzyme A is nothing but acetyl coenzyme A will convert to carbon dioxide and water. How? We'll discuss one by one, okay? Then the first one, acetyl coenzyme, has been formed. This is the first most important element by pyruvate dehydrogenase. This is very important. NADH has been formed, and as a decarboxylation, what we call oxidative decarboxylation. Then acetyl coenzyme A, in combination with oxaloacetate, a 4-carbon compound, under the influence of an enzyme citrate synthase, will form citrate, a 6-carbon. Acetyl coenzyme A is a 2-carbon, oxaloacetate is a 4-carbon. So combination leads to 6-carbon. The citrate, the first tricarboxylic, then it becomes cis aconitate through enzyme aconitase by removing water. I told you now, acetyl coenzyme conversion to H2O and CO2. We'll discuss how. Then this cis aconitate become isocitrate. Again, the acting enzyme is aconitase. Here what is removed. Then isocitrate become oxalosuccinate by isocitrate dehydrogenase. Here NAD plus form. They are called, I told you, important electron transport. NADH plus H plus. Then this again become, by removing one carbon, that means under the influence of an enzyme isocitrate dehydrogenase, become alpha keto glutarate. It is 5-carbon. Till that it is 6-carbon. It becomes 5 simply by decarboxylation. That means one carbon has been fallen off, as removed away. Then it becomes succinyl coenzyme A. On base of an enzyme alpha keto glutarate dehydrogenase. Again here one more carbon has been removed because its succinyl coenzyme becomes 4-carbon, and NAD H plus will be formed. H plus, and coenzyme A compound, that means SH, will be formed. And the succinyl coenzyme is a 4-carbon compound. Become succinate by succinate thiokinase. Here coenzyme A, SH will be removed, and GTP will be formed here. GDP plus PI leads to GTP. And this succinate become fumarate. Base of enzyme succinate dehydrogenase. Here FADH2 is formed. Then this fumarate become L-malate. Under the influence of simply by giving H2O, enzyme fumarase. And this L-malate become oxaloacetate by malate dehydrogenase. NAD plus, again it form NADH plus H plus. Before going to discussion, we have to focus, I told already, How the ATP is produced in this entire what we call glucose oxidative energetics, simply by NADH. Apart from the substrate level phosphorylation, how the ATP is formed, what we call oxidative phosphorylation by involvement of reducing enzymes like that means coenzymes NADH and FADH2. That means nicotinamide and dinucleotide and flavonoids and dinucleotide. Its reducing forms will involve the electron transport system. That means they are the electron donors, and the coupling with oxidative phosphorylation they form ATPs. So how many number of NADH and FADH2 formation is most important point, because they connected with ATPs. I told you already, one NADH leads to 3 ATPs and one FADH2 leads to 2 ATPs. So it is standard, you know. Then we have to count. Here one NADH is formed because the pyruvate become acetyl coenzyme A simply by another the ends of enzyme pyruvate dehydrogenase, and D is the other way is called this is oxidative decarboxylation. Then another one here is formed when isocitrate become oxalosuccinate, that means and the presence of isocitrate dehydrogenase, another one. Then alpha ketoglutarate, when it becomes succinyl coenzyme A, another NADH is formed. If the carbon dioxide removed, that means fifth become the fourth one. Then here when succinate become fumarate, one FADH2 is formed. Then when malate become oxaloacetate, the fourth one NADH is formed. That means totally 4 NADH and 1 FADH plus 1 ATP, what we call substrate level phosphorylation. You know, when succinyl coenzyme A becomes succinate, when a GTP is formed and they readily converts into ATP. That means the output, what we call the energetics, is this 4 NADH and 1 FADH and 1 ATPs. You know, in the discussion, in the earlier of the discussion, I told you why it is called tricarboxylic acid cycle. The first carbon, this is 6 carbon citrate, this tricarboxylic. This aconate, this tricarboxylic. Associated, this tricarboxylic. Then citric as the first component. And Krebs cycle, you know, here there is a combination of acetyl coenzyme and oxaloacetate. And how they undergo different reactions, you know. The 6 carbon first become 5 carbon by means of alpha ketoglutarate. This is very important. 5th carbon, then again become 4 carbon, that means succinyl coenzyme A. That means then again it will form oxaloacetate. That means there is a continuous regeneration and recycling of oxaloacetate. This is very important. This is how the Krebs cycle has been sustained, and this is how our ATPs has been produced in a continuous manner without the regeneration and recycling of oxaloacetate, which is what we call catalytic activity or catalytic activator. That just like an enzyme, a Krebs cycle is not possible because it is involving in 65 to 70% of the ATP production in our body. That means major energy production is coming out of it. This is very important. Okay. And these NADH and FAD, they will go to the electron transport system which is nearby. That means all this happening in, you know, matrix. All this happening in mitochondrial matrix. Then the electron transport system. Okay, this is very important. You know. Then we will discuss about the energetics of Krebs cycle. Regarding energetics, you know, I told you already how they will form. This is a very important point. Energetics regarding Krebs cycle. You know, I told you already there are, you know, 4 NADH. Actually, some scientists considered pyruvate compound as separate. So we will go as per the conventional pattern. That means 3 NADH. 3 NADH will produce 9 ATPs. Then 1 FADH2. 2 ATPs. Then 1 substrate level phosphorylation. Totally 12. That means when 1 acetyl coenzyme A is completely oxidized, form 12 ATPs. This is very important point. You know, and one more important thing is, you know, for example, you know, The end result I will tell you. C6H12O6 plus 6O2, plus 38 ADP plus 38 PI, it generates 6CO2 plus 6H2O plus 38 ATP. This is what we call the central theme. When one glucose molecule is oxidized, then it will produce 38 plus 6CO2 plus 6, what we call here oxygen, that means carbon dioxide and water. When glucose become oxidized, they produce here potassium and water. And regarding its some of the basic compounds, 2780 kilojoules of heat will be produced when one glucose molecule burnt, out of which 1590 kilojoules has been trapped in the form of 38 ATPs, and the rest of that means just 48% have been utilized, and the rest of the energy is waste. This is a major energetics. That means out of when one glucose molecule is burned, 2780 kilojoules have been releasing, but out of it only 48% have been utilized. That means in the way of 1590, that means equal to 38 ATP. That means 1 ATP equal to 30.5 kilojoules. This is very important. That means at the rate of 48%, the rest of the energy is going waste. What we call enthalpy. Okay, we'll discuss in the alternate transition regarding other issues. Okay, and thanks for listening. You can